All right, welcome to this highly technical visual dossier. Today, we are cracking open the master files of a seriously groundbreaking project. We're looking directly at the declassified open-source blueprints engineered by Lázaro López Paz for the creation of next-generation smart peptides. This explainer is going to be a deep dive into the raw data. We are stripping away all the fluff to focus entirely on the algorithms, the physical chemistry equations, and the precise laboratory protocols that take these molecules from quantum simulations into physical reality. If you want to understand the absolute bleeding edge of human enhancement biotechnology, you are in exactly the right place.
Okay, let's quickly look at our roadmap. We're going to cover 1. Computational foundations, 2. Physical-chemical equations, 3. Transition to practical viability, 4. The smart peptide catalog, 5. The SPPS protocol, and finally, 6. Roadmap and budget.
So, starting off with Section 1, Computational Foundations.
Looking at the first page of the Master Manufacturing and Integration Dossier, version 3.2, you can see the philosophical grounding right at the top. The development of systems like neuroplastin isn't just about precision pharmacology. It's fundamentally aimed at expanding cognitive capacity and integrating the human neural network with far greater fidelity. But, the physics presents a massive hurdle. Simulating a 20 amino acid peptide with its full salvation environment from scratch literally causes a complete breakdown in current quantum hardware. It's just too much data. So López Paz designed this brilliant hybrid strategy, merging quantum mechanics and molecular mechanics. The standard peptide backbone is modeled using traditional molecular mechanics, while the critical photoactive center is completely isolated and processed using quantum estimators.
And here are the exact mathematical formulas driving that quantum isolation. First up, we have the variational quantum eigensolver, or VQE equation. H applied to the state psi of theta equals E of theta times the state psi of theta. This gives us the exact energy state of the photoactive center. But listen, it's not just about activation, right? To ensure the molecule actually returns to its resting state and remains totally stable at human body temperature, we rely on the Arrhenius thermal relaxation equation below it. That's K equals A times E to the power of negative E sub A divided by R times T. This isn't just theoretical math. It's the absolute prerequisite for human application, ensuring the structural integrity of these peptides at exactly 37 degrees Celsius.
Moving right along to Section 2, Physical-Chemical Equations.
Now, check out this exact photopharmacology kinetics equation pulled directly from the dossier. We are looking at the rate of change of the trans isomer over time. d trans dt equals negative phi subtrans times I0 times 1 minus 10 to the power of negative A plus K sub cis times the concentration of cis. The absolute crucial variable in this entire equation, I0, the intensity of the incident light. By calculating this exact kinetic threshold, the researchers figured out that applying standard commercial LEDs at 365 nanometers is perfectly sufficient to force the trans to cis isomerization. Basically, that specific light intensity acts as a switch, effectively turning the peptide on or off right inside a living cell.
But of course, a smart switch is completely useless if the peptide doesn't actually bind to the right target. To design the peptide backbone, Lázaro López Paz utilized RF diffusion 2 algorithms paired up with protein MPNN. Here is the exact AI affinity loss function they used to optimize the sequences. It balances out shape, interface, clashes, and salvation. But pay really close attention to that final variable, L sub n CAA. That right there is the critical factor. It trains the model to accommodate non-canonical amino acids, like our late-sensitive azo-F, ensuring the folding remains completely stable, even when we introduce these artificial components.
Which brings us to Section 3, Transition to Practical Viability. Let's contrast version 2.0 against the finalized version 3.2. Version 2.0 is a computational masterpiece for sure, but honestly, it was an absolute nightmare to actually manufacture. It relied on complex DNA origami vectorization and custom synthesized polymers that maybe a handful of labs in the entire world could reliably produce. Version 3.2, however, strips all of that away in favor of real-world viability. DNA origami gets replaced by standard cell-penetrating peptides, custom polymers swapped for commercial MPG malamide, and the highly custom photoactive amino acids are entirely replaced by commercial azo-Fef, which you can literally order right out of the Sigma Aldrich catalog. This pivot is a massive game-changer. It means that any standard university or CRO lab in the world can synthesize these smart peptides today.
Let's dive into Section 4, the Smart Peptide Catalog.
This is it. the raw chemical catalog. These aren't just theoretical concepts anymore. These are highly actionable formulas. Take neuroplastin version 3, designed specifically to stabilize the GluA1 AMPA receptor for enhanced synaptic plasticity. The exact sequence reads acetyl YGRKK RRQRR followed by a GSG linker, then GRK, then our photoactive azo F center ending in RLYSS with an amide terminus. Or look at senolytic X version 3. This is a photo-controlled senolytic designed to clear aging cells by disrupting the FOXO4P53 interaction. Its sequence starts right out of the gate with acetyl-lysine bound to azo-F, followed by the penetrating motif and the binding sequence. What we're looking at right here is the bleeding edge of actionable biotechnology mapped out letter by literal letter.
And this brilliantly illustrates the true value of the dossier. The exact sequence catalog we just discussed is mapped directly to the master protocol for manufacturing. The blueprint doesn't just give you the what, it meticulously hands you the how, linking every single formula straight down to the laboratory floor.
Section 5. The SPPS Synthesis Protocol. So how do we build it? The 0.1 millimole solid-phase peptide synthesis process starts with swelling the rink amide resin in DMF for 1 hour to prep the support matrix. Next comes your standard cycles, deprotecting the FMO group with 20% piperidine, followed by coupling the standard amino acids using HATU and DIPE. But Step 3 is the absolute linchpin. The insertion of the azopho photoactive center requires a double coupling over 3 hours, and it must be done in absolute total darkness. Vials wrapped in foil, working only under dim red light. We have to ensure the molecule doesn't prematurely isomerize. After that's done, you cleave the peptide from the resin using a TFA cocktail, precipitate it in cold ether, and finally, run it through preparative HPLC and mass spectrometry to guarantee a purity of over 95%.
Of course, successfully synthesizing the peptide is only half the battle, right? It has to be biologically safe. The dossier explicitly outlines some very rigid safety constraints. Because we are using highly effective cell-penetrating peptides, CPPs, in vitro working concentrations must be kept strictly below 20 micromolar. That prevents indiscriminate cell membrane damage. Furthermore, the protocol mandates conjugation with commercial MPEG melamide. This acts essentially as a steric shield around the peptide. And why does this matter? Well, it severely reduces nonspecific immune responses and prevents the body from clearing the peptide out prematurely before it even reaches its target. Finally, for any in vivo application, final batch endotoxin levels must be rigorously verified to be less than 1 endotoxin unit per milligram. These parameters are absolutely non-negotiable for lab viability.
Finally, Section 6, Roadmap and Budget.
800 to 2,500 euros. That is the estimated academic budget required to synthesize just one of these priority peptides, like your neuroplastin or Senolid X, from scratch all the way up to a pure state, ready for cellular assay. That covers the standard amino acids, the commercial azofuf, the resin, the coupling reagents, the whole shebang, even the HPLC purification. It is a stunningly accessible price point for frontier biotechnology, and it's made possible entirely by the pragmatic redesign of version 3.2.
And to ensure this open-source blueprint remains protected and actually viable for future clinical development, the dossier even includes provisional patent frameworks. The claims are incredibly precise. Claim 1 locks down the exact version 3 sequences combined with the photoresponsive non-canonical amino acids. Claim 2 protects the specific PEG polymer conjugations we just talked about for safety. Claim 3 covers the actual therapeutic applications, like controlled senolysis and synaptic plasticity. And Claim 4 protects the specific physical-chemical methods of activating these molecules using exact light wavelengths between 365 and 660 nanometers.
So we've unpacked the quantum algorithms, we've decoded the loss functions, we've read out the exact chemical sequences, and walked right through the manufacturing floor. The Lázaro López Paz blueprint has successfully bridged the massive gap between computational theory and accessible laboratory reality. The science is no longer locked away behind the doors of billion-dollar pharmaceutical giants, the formulas are right here. So the ultimate question is this. With the design, the exact mathematical sequences, and the step-by-step protocols now directly in your hands, how will you use this dossier to push the boundaries of human enhancement in your own lab?